Hello everyone, I'm Sheikh Arif from India and I'll be presenting my team's work on transcriptome gene expression data retrieved from Cancer Atlas genome. Our team comprising of Ms. Sanjida Akhtar, Sheikh Arif, that is me, Meena Zakaria, and Benedict Ajunku. In the field of cancer research and treatment, transcriptomics is playing a very large role, primarily by identifying biomarkers which can detect cancer very early. Two, by understanding the molecular pathway which are driving the cancer. And number three, by having gene expression profile which are enabling us to precisely classify tumors and guiding us to personalized treatment and drug development. So the primary objective of this paper is to analyze gene expression data retrieved from Cancer Atlas genome to identify gene signatures that are involved in cancer. The methodology that this paper resorts to is of collecting more than 4,000 cancer samples and 548 normal sample data across 21 different types of cancer from Cancer Atlas genome. Then it goes into differential expression of gene of the transcriptome. Then it further goes into clustering those gene. From the clustering of data, it goes into gene set association analysis. Then it looks for gene signature across different type of cancer. Then it particularly identifies gene signature in specific type of cancers. This is the overall workflow of the paper where different bioconductor tools were used for differential gene expression analysis and for further pathway enrichment, gene ontology and kick pathways were used. Coming to the key findings of the paper, the researchers were able to identify 14 gene signatures in 7 different types of cancer. Number 2, they were able to identify 7 cross-cancer gene signature which were at least altered in 4 types of cancer. Number 3, they were able to identify genes which are related to cell cycle which were highly expressed in comparison to other type of genes, showing the fact that genes related to cell cycle are involved in cancer. Number 4, they were able to identify specific gene signatures to lung cancer. Number 5, to have a valid and robust study, they validated their study on non-cancer atlas genome dataset so that they can use their findings in cancer treatment and diagnosis. So our final interpretation of the paper is that the genes that were found to be deregulated in cell cycle process can be targeted for proper development of cancer care. Number 2, gene signature that were validated using independent dataset can be helpful in distinguishing cancer from normal tissues. And most importantly and last, that this finding has the potential for developing diagnostic tool for personalized therapy. The challenge that lies ahead of us is to find a link between the mutational profiles and the differential expression profiles in cancer cells. The scope of future research is to find a relationship between mutational and differential expression profile and to find out how gene expression pattern changes in vicinity of driver mutations. This is the reference of the paper which we have presented. That's all from me and my team. Thank you.